we're now transitioning to another set of the sensory system. So we spent 2 lectures talking about the visual system. It's a very complex system. The sensory organ, the eye, is very complex. And there's a lot of very interesting processing that happens within those systems. We're now going to discuss 2 other sensory systems all in one lecture. They share some similarities, but obviously they're also very different. And we will be talking about these systems all in one lecture because they're relatively more simple, and there's also a little bit less than we understand about these systems than we do about the visual system. So today we're talking about the chemical senses. That is the main commonality that it is that these 2 systems have, and we're going to be talking about the sense of taste and the sense of smell. But before we get into that, I wanted to discuss here in this lecture the facial nerves. So this is a very important concept within understanding the nervous system, and so we're going to cover this in some detail right here, and we won't be going over this thoroughly. But it is something that is critical that you are familiar with and something that you understand. There are 12 major nerves that leave the central nervous system in the head and innervate various parts of the face. These nerves they use Roman numerals to describe them, and it goes from 1 to 12. We won't be talking about all of them, and I don't necessarily expect you to memorize all of them. You do need to know the nerves that we talk about, and you should know which number those nerves are. Okay, so I do expect you to have memorized the number for the nerves that are going to be most important that we're going to be talking about. Now, the nerves can be broken into 2 main categories. There are sensory nerves, and then there are motor nerves. The sensory nerves obviously sense the world, and then there are motor nerves that control the muscles. There are some nerves that are mixed as well, but primarily you're going to find that many of the nerves are specialized into one or the other. We have talked so far about the visual system, and the main nerve there is the optic nerve, which goes here into the optic chiasm. The optic nerve is connected to the eye, and this is known as the second facial nerve. Okay, so this is facial nerve number 2, the optic nerve. We will be talking about 3 nerves that are critical for taste. The facial nerve, which is nerve 7, the glossopharyngeal nerve, which is nerve 9, and the vagus nerve, which is nerve 10. All of these innervate the tongue. And then the second half of the lecture, we're going to talk about the olfactory system, and that is nerve number 1, the olfactory nerve, going from the olfactory bulb to the brain. So, those are the 3 nerves that these are the 3 categories of nerves. Okay, so let's talk about taste. Humans recognize 5 basic tastes. We have saltiness, sweetness, sourness, bitterness, and umami. These are self-explanatory, but umami is a relatively new taste. Everyone for centuries has known that we could have these 4 basic tastes, but umami was one that was recognized as a very specific category. It was originally identified by the Japanese, and they're the ones who named it. So it has this Japanese name, umami, which means delicious in Japanese. And it provides, there's a specific subset of taste buds that have umami receptors on them that help us to detect the savory taste of amino acids. And in particular, it detects glutamate. And I'm sure you all have heard about MSG and how bad it is. But MSG is just monosodium glutamate. It is essentially a glutamate salt. And so the debate about how bad MSG is complex, since it is basically just an amino acid. But the reason why that amino acid is added to foods is because it gives a very savory, meaty kind of taste. Okay, so we got 5 basic tastes, but there are all kinds of different tastes out there. When you eat pizza or lemon ice cream or bubble tea, any of these things, you know, there's a lot more than just 5 of them, right? So with this limited number of basic tastes, how do we perceive the countless flavors of food and drink? Well, that's because foods activate distinct combinations of taste receptors. So for instance, lemon ice cream, you're going to have a very strong combination of sweetness plus sourness that gives it its unique set of tastes. The flavors are also combined with the smells as well, and smell helps to give us the combination of sensory inputs that gives us the distinct experience of particular tastes. Lemon also has its own unique smell, as does pizza. And so those things also help color what we describe as taste. Texture and temperature also contribute. So the sensations of pain, which we will talk about in a bit, in the next lecture, I think. Yes, next lecture. So having spicy flavors such as hot sauces, those actually are not acting directly onto the taste receptors, at least not exclusively. There are also pain receptors that help carry the information of being spicy or minty or other things as well, which contribute to this process as well. We're not going to talk about those today. We'll talk about those in the next lecture. So taste is primarily a function of the tongue. There are other regions within our mouth that also have taste sensitivity, but the tongue has the vast majority of them. So taste sensing cells are found in what are called taste buds that are distributed on the surface of the tongue and these other regions. So most taste stimuli, they're called tastants, are non-volatile and they're hydrophilic. So these are things that like to be diluted into water and they don't just float around like odorants do that we smell. So these are going to be soluble in saliva that allow us to process what we're actually tasting. So it has to be able to be put into liquid format. The perceived intensity of taste is usually directly proportional to the tastant concentration. So the more salt that you have on something, you're going to increase the concentration of salt, which is going to make us perceive things to be particularly salty, for instance. Okay, many of us have this impression or have grown up with the idea that we have very distinct regions of our tongue that are specifically directed towards particular tastes. So in the back, we have this assumption that that's where bitter taste is, sour's on the back side, salty's on the front side, and then sweet right up at the front. Now, it is true in general that there is sort of regional increased sensitivity, as you can see here, so that it is true that sour is generally going to be better detected on the sides of the tongue than in the middle, bitter towards more in the back. But it is not true that that's where these receptors are exclusively found. In fact, the receptors for these 4 different tastes, 5 tastes, are found all across the tongue. It's just that their density tends to be a bit higher in certain parts of the tongue. So if you touch something that's bitter towards the tip of your tongue, you will taste it there, and it's not because it's slipping towards the back of the tongue. It's because there are bitter receptors located towards the front of the tongue. Same for salt. You put that towards the back of the tongue. There are salt receptors all the way in the back of the tongue. It just so happens that the higher density of those salt receptors are located towards the front. So there aren't distinct regions where you're only going to find that sensitivity, and in fact, you'll find sensitivity across the whole tongue. It's just that the density tends to differ depending upon where you're talking about on the tongue. Tastings are detected in receptive specializations called papillae, which contain taste buds and taste cells. Okay, so we're going to talk about these papillae a little bit. So papillae is the plural form, papilla singular. So a papilla has 1 to several hundred taste buds that contain neuroepithelial taste cells. So let's see, this is what a papilla looks like right here. It's these kind of big bumps on the tongue. That's a papilla there, that's a papilla there. But if you zoom in and look, what you'll see is, yes, there's this bump, but then there's this ridge that actually is like a deep canyon that goes down into the tongue. And then if you get closer and zoom in even more, you can zoom in on what then is an individual taste bud. So this is what the taste bud looks like in cross section. Taste bud will have a taste pore, several different taste cells, and then there are additional cells called basal cells, and then gustatory afferent axons. And we will talk about the organization of these different taste cells across the different tastes in a little bit more detail across the next several slides. Now, each person has around 2,000 to 5,000 different taste buds, but there's actually a huge range of variability across individuals. There's some people who have really poor senses of taste. Those individuals tend to have fewer taste buds, as many as 500 taste buds across their entire mouth. But there are some people who are super tasters. They can have as many as 20,000 taste buds. That's a lot. So there's a huge range, but on average, it's somewhere between 2,000 to 5,000 taste buds. So these taste buds have an apical end, which is located here, and that has the taste pore, and that is what interacts with the saliva. So saliva comes down into these channels, and then the chemicals that are within the saliva will interact there at the taste pore, where the taste cells have little microvilli that attach, that are interacting with the chemicals there. Basal cells are the stem cells that are continually dividing to replace taste cells. So your taste cells actually have a life cycle. They're present for some time, and then eventually they just die. So the taste cells are not neurons. You can just look at how they look. You can see they're definitely not neurons. Sort of like the photoreceptor cells. Taste cells, in fact, are epithelial cells, sort of deriving from the same parts of the embryo that skin does. So is the nervous system. Move on. The taste receptor cells, they form synapses with the endings of gustatory afferents near the bottom of the taste bud. So they have this kind of bulb-like shape, and then they have these synapses down near the bottom. The type of neurotransmitter that you can find at any given synapse is going to be dependent on the specific stimulus type that that particular taste cell is sensitive to. Okay. So sour and salty taste cells, they use serotonin as its neurotransmitter to interact with the primary gustatory afferent neurons. So it's serotonin that is released from salty or sour neurons. The other 3 taste cells, sweet, bitter, and umami, they use ATP as a neurotransmitter that acts on to those gustatory afferents. Neurotransmitters released onto the within the synapse onto the gustatory afferent, and that causes that that sensory neuron to fire an action potential. So that's saltiness. It's a pretty straightforward process. It's simply driven by changes in sodium concentration. Sourness uses a somewhat similar mechanism. So sour tasting food is due to high acidity. It's very acidic. Acids have low pH, and pH is determined by the imbalance of free hydrogen ions. So with more free hydrogen ions in a solution, the more acidic it's going to be. So let's say you eat some of that lemon ice cream, there's the relatively acidic citric acid. That's going to include a relatively high level of free hydrogen ions. That gets into your saliva. And so then from there, hydrogen can affect taste receptors by binding or blocking potassium channels. So that'll block the potassium channels, but then it also includes going through proton channels. So then you can have this depolarization that happens by the potassium channels being blocked and hydrogen flowing through, sort of like sodium being positive. So you have this cation flowing through. This leads to membrane depolarization, which from there then causes the same basic mechanism that's found in salt cells, with an increase in intracellular calcium, leading to the vesicles that are filled with serotonin neurotransmitter to bind to the cell membrane of the sour taste cells, leading to serotonin release, causing the postsynaptic gustatory axon to have an action potential. Pretty similar and simple mechanism. All right, from here, now we're going to talk about the three other categories of taste cells, and they have somewhat more complex mechanisms. It isn't simply just differences in ion concentration. So bitterness, sweet, and umami, they are sensed via dimers of G protein coupled receptors. We refer to them as T1R and T2R proteins. And so they have these dimers that'll give you a complex set of more specific receptor types. So with bitter, there's actually 25 different types of bitter receptors in humans. And so one of their major functions is to act as poison detectors. Poisons have certain compounds in them. We have evolved a wide variety of these T2R proteins to allow us to detect a range of different things that we refer to as bitter. Bitter, a lot of people don't care for bitter. At least bitter is sort of an acquired taste in many cases, such as like coffee. Kids will tend to not like coffee, and you have to kind of grow to appreciate coffee as an adult. And that's because there's all kinds of bitterants that are in there. And in fact, like raw coffee has all kinds of poisons and toxins for other animals. For humans, it's not so bad. So the process then is that bitter tasting foods, there'll be some kind of protein or compound that'll bind to a particular T2R G protein coupled receptor, this dimer. This stimulates the enzyme phospholipase C, PLC, right here. So this G protein coupled mechanism, you get to a secondary messenger system that increases the production of IP3. IP3 triggers the release of intracellular calcium stores. So we've got calcium stores, endoplasmic reticulum typically, induces release of calcium into the cytoplasm. The calcium opens up sodium channels, and then this calcium, so then it leads to further membrane depolarization. But the calcium also then will trigger then the release of ATP, and then ATP acts as a neurotransmitter at these synapses, where it acts on ATP-specific receptors that then causes an action potential in the afferent. Okay. Sweetness. Sweet tastants are natural and the artificial forms. They're detected by the same taste receptor protein. So with sweetness, we have a much more simpler family of taste receptors. They are also dimers of G protein coupled receptors, but we only have two T1R receptor types. We have T1R2 and T1R3. If either of these are present, then these are going to be sweetness receptors. And if there's a mutation in one of these, then or if it's missing, then sweetness may not be perceived. You have to have one of these two in order to detect sweetness. Sweet tastants, such as glucose, for instance, or sucralose or sucrose, fructose, they will bind to this G protein coupled receptor, to the dimer. And so, if you, like I said, if you have a mutation in one of these or one of them is missing, you can't detect it because you can't make the dimer. This leads to the activation of the PLC, increases IP3 production, triggers the release of intracellular calcium stores. Calcium stimulates ATP release to activate the postsynaptic purogenic receptors, which is sensitive to the ATP. So it's very, very similar processing. And obviously we have similar proteins that enable us to detect bitter and sweet tastes. So how do we then not confuse bitter tastants from sweet ones? It has to do with the specific receptor protein. So the receptor proteins that is taking the sensory perception, this reception and the transduction side is allowing for the taking chemical information in the environment perceived by these particular receptors, and then those particular receptors are found in different taste cells. We have a sweet taste cell and a bitter taste cell. You won't have a taste cell that expresses both bitter and sweet. So the differences that we can see between bitter and sweet is that they are actually in different taste cells. Okay, and then they connect to different gustatory axons. So the transduction of this information into the brain then goes down very separate pathways. You will have a sweet pathway and a bitter pathway, and it keeps those. UMAMI, so let's walk through UMAMI a little bit. These receptors also involve a G protein coupled receptor mechanism. It requires T1R1 and T1R3. So you need to have this G protein coupled receptor dimerization. And it has a very similar mechanism. So the amino acids such as glutamate will bind to this UMAMI receptor, activating a PLC mechanism, leading to increase in IP3 production. This triggers the release of calcium stores, and calcium stimulates the ATP to act on the postsynaptic cell. So how do we not confuse umami, bitter, and sweet? Again, the receptor proteins for each type are expressed in different taste cells. So you have umami taste cells, bitter taste cells, sweet taste cells. They all have a very similar internal molecular mechanism, but the difference is these G protein coupled dimers. We have two different kinds of dimers. It allows for the specificity of the kind of tastant that it's going to perceive, and each taste cell has its own, only one particular combination of G protein coupled receptors. Okay, that's the difference. All right, so then how do we get how do we get transfer of this information up into the brain? As I mentioned before, we have three facial nerves that innervate the tasting areas of the mouth. We have the facial nerve, which innervates the first 2/3 of the tongue. So the facial nerve is the seventh facial nerve. The glossal pharyngeal nerve, which is cranial nerve 9. Cranial nerve 9 innervates the back third of the tongue. And then we have the vagus nerve, which innervates the epiglottis, but there are all kinds of taste receptors back there as well. So the vagus nerve, the glossopharyngeal nerve, and the facial nerve, these are all carrying the 5 different tastant information up into the central nervous system. Each of these nerves synapse, the ones that are carrying the sensory information, they synapse onto the nucleus of the solitary tract. This is also known as the gustatory nucleus. So this nucleus is really the first main station within the central nervous system that's receiving this taste information, and then that ascends up into the brain, synapsing onto the ventral posterior medial nucleus of the thalamus. So it's now carrying this information to the thalamus, which we described before as being this relay station that's critical for carrying sensory information to the cortex. So, and then this is showing again the synapses. So this part is shown here. So these are the ascending pathway from the nucleus of the solitary tract, synapsing onto the ventral posterior medial nucleus of the thalamus, okay? So the ventral posterior nucleus of the thalamus, then these neurons synapse onto the insular taste cortex, and they also will synapse onto the frontal taste cortex. So two different brain areas, the insula and in the frontal cortex as well. And those are going to be the main initial parts of olfactory cortex. Okay. So this is a very simplified schematic of what this looks like. There are parts of this that we are not going to talk about in detail now. Instead, we will talk about them when we talk about these brain areas, such as the hypothalamus and the amygdala. But this includes some of these important parts, right? So the first four stations that we have the taste buds on the tongue, sensing the taste information. It is carried on these three cranial nerves, the facial, glossopharyngeal, and vagus. That information is carried up to the nucleus of the solitary tract, also known as the gustatory nucleus, up to the ventral posterior portion of the medial thalamus, or VPM of the thalamus. And then that connects to the insula in the temporal lobe and the frontal cortical areas that are olfactory cortex. Okay. Then from there, that taste information is then processed by other brain areas, especially the amygdala and the hypothalamus. But like I said, we will cover those when we get to that. Gustatory cells, they also project to other brainstem regions involved with swallowing, gagging, and vomiting. This is more instinctual circuits that allow you to throw up. So if you have something in your mouth that's particularly gross, these circuits will be activated and cause you to throw up. So I will let you discuss, try to figure out what are the four basic steps for sensory pathways in taste. Again, sensory reception, transduction, transmission, and perception. And I want you to be able to do and describe all of these four basic steps in the sensory pathways for all of the sensory pathways that we're going to be talking about. So if we talk about what sensory reception is for vision, taste, smell, touch, or hearing, I want you to be able to give some detail about what each of these different steps are and know what are the similarities and differences between each of the sensory systems. All right, that is taste. Now we're going to switch over to smell. Smell, your sniffer, your nose. It's actually a very interesting and I think a relatively complex system. It's actually quite elegant, quite beautiful. It's one of my favorite sensory modalities. And I hope that you'll grow to appreciate it just as I do. You know, it's the only way that we can, when you smell the roses, there's actually a lot of beauty happening inside your head to make that happen. That's very poetic. Okay, so we have the basic function of smell is you inhale air. It's going to be filled with all kinds of chemicals called odorants. And then it's going to touch upon what's called the olfactory epithelium. So our nose itself, the part right here that you can kind of see and just on the inside, that isn't what is doing any of the smelling. Instead, the air actually has to go up quite up inside the nasal cavity, and it interacts with the olfactory epithelium, which in humans is located on the top part of the nasal cavity. So the olfactory epithelium is a very thin sheet of cells, and it's comprised of 3 different cell types: olfactory receptor cells, and these are actually neurons. So unlike taste cells and photoreceptor cells, olfactory receptor cells, in fact, are neurons, and they transduce the olfactory signal and send that information into the central nervous system. There are also supporting cells. These supporting cells act very similar to glia. But they also produce mucus to keep, to create this mucus layer at the surface of the epithelium. And then there are basal cells, and these are basal cells that are very similar to the kinds of basal cells you find in the taste system, in that they are stem cells that also produce new receptor cells. So these basal cells will divide and give rise to new receptor cells. So there's turnover of receptor cells as well, just like the taste cells. That connects to the olfactory bulb. Go through this again. Sorry, too fast. So you have membrane depolarization, which then, as long as there's enough odorant receptors that are activated, leading to an action potential in these olfactory receptor cells / neurons. Okay, so we talked about the taste receptors that, like sweet, there's basically one combination of the dimer G protein coupled receptors, same for umami. Bitterness, there's a little bit more variety. You have 25 different kinds, giving you a little bit more complexity to the bitter tasting, right? So there's, you can imagine there's a wider variety of chemicals that we can pick up and perceive as bitter. Olfactory receptor proteins blow all of those out of the water. There's a huge range of olfactory receptor proteins. And each individual neuron will express a single olfactory receptor protein. Each olfactory receptor neuron is going to have a particular odorant receptor that it expresses, which is why we have these different colors here to represent a particular receptor that each neuron is expressing. The number of olfactory receptor proteins varies enormously across species. Animals that are particularly olfactory sensitive, such as rodents, they have more than 1,000 different receptors that they express. In fact, if you think about each receptor has to be expressed, that information has to be contained in the genome. Nearly something like, I know in rodents it's something between 1,000 and 2,000 of the genes that make up the rodent genome, which is 30,000 genes. Those are just odorant receptors. They have a huge range of odorant receptors that they express. Humans, we are not particularly olfactory centered, so we have a lot less than rodents. We have around 350 odorant receptors in our genome. So because each olfactory receptor neuron expresses a single olfactory receptor protein, that gives us then a specificity of that particular neuron being fired. But we have all kinds of different smells that we can pick up, right? And again, that has to do with the combination of the different kinds of receptors that neurons that have been activated. So the olfactory receptor neurons will fire. They don't necessarily have super high specificity to a single chemical. Some of them do, but many of them will pick up different kinds of chemicals, and they'll have differing levels of sensitivity depending upon the chemical and how much of the chemical is there. So for instance, let's say we're looking at this particular green olfactory receptor neuron. You can see that this neuron particularly likes citrus, the citrus chemicals. It kind of likes floral chemicals. It has a few action potentials, but not a lot. It also seems to like peppermint, but it doesn't respond to almond at all. The red neuron, though, doesn't like citrus, but it's particularly sensitive to almond, and it also happens to be sensitive to peppermint. So you can see that, okay, we don't have super high specificity, but instead, because different chemicals are going to act on different olfactory receptor neurons in different ways. It is the combination of the response that'll tell you which odor that you're smelling. And this is what's referred to as a population coding scheme. So if we are smelling citrus, you're going to have these green neurons that are firing an awful lot, a little bit of the blue, and none of the red, and that will tell you, hey, there's some citrus in here. If we have almond there, we're not going to have any green, tiny little bit of the blue, but the red responding quite a bit. That will tell you that that's almond. And it'll be this combination, this population coding scheme, that tells us, hey, this is almond or citrus or both, right? So I have a little bit of the history of this and how this was discovered. Richard AXEL and Linda Buck, they were both winners of the Nobel Prize in 2004. And it was Linda Buck, she was working as a postdoc in Richard AXEL's lab. She discovered, she and him, as well as other people, but they discovered the family of odorant receptors. And they worked out a lot of these details of that different olfactory receptor neurons express just a single odorant receptor, but that there are hundreds and hundreds and hundreds of different odorant receptors that are found. And they also worked out some of the details of that G protein coupled mechanism as well. That got them the Nobel Prize in 2004. Central olfactory pathways then. So this is another part of the system, and I find particularly elegant. It's actually quite beautiful to look at in brain slices as well. So let's walk over it. So again, we have the olfactory epithelium right here at the top of the nasal cavity. And you've got all your olfactory receptor cells in the olfactory epithelium. There's the little cilia there. So let's say an odorant receptor neuron starts to fire, and here's a group of them that then send their axons up to the olfactory bulb. So now this is where we have our first part of the central nervous system getting involved. So these axons connect to the olfactory bulb. That's our first order neuron. And then from there we have second order neurons. There are neurons on the other side of that that then send their axons up into the cortex, in fact. So this interaction is happening at what's called a glomerulus. And we're going to look at a little bit more detail of what's happening in a glomerulus and learn a little bit more about these secondary order olfactory neurons on the slide. There is specific mapping of olfactory cells and receptors onto specific glomeruli within the olfactory bulb. So the olfactory bulb on the outer surface, it'll be covered with all these little circles called glomeruli. Glomeruli is plural, glomerulus is singular. Each glomerulus is receiving input from olfactory receptor cells that express only a single receptor protein. So these blue cells, they have one particular olfactory receptor protein that they're expressing, and all of that information is connected into a single glomerulus. So you can see we're now sort of gathering the sensory information and organizing it into an olfactory receptor specified way. So the blue receptor neurons are connected to this glomerulus. Red is over here and green is over here, and this is referring to the different kinds of receptors that are expressed there. Another note on the olfactory bulb, so just as the rat olfactory epithelium has a very large representation in its nose relative to its brain compared to humans, this is also reflected in the olfactory system itself. And the olfactory bulb in rats is huge. It makes up something on the order of about 20% of their entire brain, whereas in humans it's less than 1%. And so this is the olfactory bulb in humans. It's super duper tiny, and yeah, so very tiny compared to rodents. So here's a Nissl stain section of a cross section of a mouse olfactory bulb. Okay, so this is, remember, Nissl labels cell bodies, and so what you're seeing are the different cell bodies. And a cross section of the olfactory bulb has, it reveals that there are different layers. So you have the glomerular layer, and that's where all the glomeruli are located. And you can see these empty spaces here. This is a glomerulus here, and that's where those axons from the olfactory receptor cells are connecting to dendrites of the second order neurons. Then we have a layer called the outer plexiform layer. Then we have a layer called the mitral cell layer. And so the mitral cells are really the secondary order axons, neurons. And then you have the inner plexiform layer, and then you have granule cells down below. And so there's a ton of these granule cells located here. Here's a schematic of how this is organized, okay? So you've got your olfactory epithelium, the olfactory receptor cells going up through the bone and connecting to the different glomeruli. And then the glomeruli are connected to primarily mitral cells. Mitral cells are the main olfactory receptor output from that's carrying the olfactory receptor information to the brain. These are the important parts that you need to know. The olfactory receptor cells, obviously you need to know those. The glomerulus, the mitral cells, and the granule cells. Granule cells are also important to this process. Right. So just so that you're familiar with what we're looking at here, this this was the dorsal side of the olfactory bulb right here. We were looking over here, and and so it's reversed compared to what this is describing here, right? So the glomerulus is down here, and the mitral cells are right here. Well, here we're looking at the glomerulus over here and the mitral cells here because they're describing it from the ventral side of the olfactory bulb, but this side is showing the dorsal side. So you need to know that, like, depending upon what side of a structure that you're looking at, you'll have to know that order. Okay, so if you're looking at the dorsal side, you'll need to know that the glomeruli are on the outside, or if you're talking about the ventral side, glomeruli are located on the bottom. Okay, top, bottom. So you got to know the order. But the order may be reversed depending upon whether you're talking about the top or the bottom, dorsal or ventral. All right, so these mitral cells, these are the critical outputs going through the olfactory neuron, which is the first cranial nerve. They connect up into multiple areas of the cortex. So they project to a wide variety of cortical areas. So this is showing in one cross-section, a bunch of different areas of cortex. We've got the piriform cortex, the olfactory tubercle, the accessory olfactory nucleus, the amygdala is located here, the entorhinal cortex. I'm listing all these names, but these are just different cortical areas that receive olfactory input, including the amygdala. And then this is showing in that same section, so this is the same section, but now it's showing in red the axons coming from mitral cells. And these mitral cells, they will split off their axons and they'll connect to all kinds of different olfactory areas throughout the brain. So the mitral cells, we learned in the previous slide, they project very widely throughout the central nervous system. This figure is showing in a little bit more schematized version of what particular areas receive input from the mitral cells. So this is the olfactory bulb here in this part. So we're looking at a human brain. Here are the olfactory receptor cells. And so this is a very schematized version. The synapse would be normally in a glomerulus, but it's just showing it projecting to a mitral output neuron. These neurons, they project to a bunch of different areas. An area called the olfactory tubercle, they project to a bunch of other olfactory cortices, including entorhinal cortex, the piriform cortex, and then as well as the amygdala, which some of you probably have heard of. We have an entire lecture dedicated to the amygdala later when we talk about fear and stress. And it'll come up over and over again. But it also receives input directly from the olfaction. One important point here that I hope that you can see is that there is a direct parallel projection to the olfactory cortex that bypasses the thalamus. So there is a connection that kind of makes its way through the thalamus, which is through the olfactory tubercle, technically this is actually part of cortex. So it goes from the sensory system, cortex, goes through thalamus, and then up to the orbital frontal cortex. But there's a ton of other information that goes straight from the olfactory system directly to cortex. This is the only sensory system that has a substantial bypass of the thalamus and connecting directly to cortex itself. All the other systems don't have a direct connection to cortex. And it is thought that this is part of the reason why smell has kind of a different salience when we talk about memory. That in some respects you get this strong sense of deja vu when you smell something. It'll bring up some kind of experience, and it'll be very different than if you were seeing or hearing something. But then there are other times when it's really hard to place that memory. That you know that you're experiencing some kind of emotion or some sort of feeling, but it's not something that you can place on a very specific event and recall the event. That often happens with smell. It is thought that part of the reason why that might be is because so much of the olfaction bypasses the thalamus and goes to cortical areas, so it's not being organized by the thalamus and gating its way through certain memory centers, and instead is dedicating itself to olfactory specific parts of cortex. Really interesting stuff. A lot of sort of mystery around it as well. So to review this again, we've got the olfactory receptors connected to the olfactory bulb, to a variety of olfactory bulb targets. Targets of the olfactory bulb, including the entorhinal cortex. And then from there, we have a number of other secondary structures. And again, we will discuss amygdala and hippocampus later. So we'll discuss like the role of smell in partner preference and how it can affect the hippocampus and that kind of thing. We'll talk about orbital frontal cortex and smells much later in the semester when we talk about these brain areas. But because I haven't described these brain areas, we can't quite get into it too much. Instead, you just kind of really need to know this pathway, that the olfactory bulb connects to these areas. Okay, so that's going to be the important part. And then we'll get to the other parts later. All right, so there are a bunch of different ways that neuroscientists can measure neural activity in live animals. And so far, just in the last lecture, you've seen two major important ways to look at neural activity in live intact animals. One that we saw was doing electrophysiological recordings of individual neurons or groups of neurons using a glass pipette or, say, an electrical wire. This gets lowered into a particular brain area, and then you can record from a particular group of neurons or even an individual neuron. We saw what Hubel and Wiesel did, recording in the primary visual cortex in cats. This is a very common technique. It is widely used, and it's been used for decades. And we will be revisiting it over and over again. The readout of this information is spikes, right? So we'll have raster plot, for instance, like we saw with the facial recognition in monkeys, and also the Halle Berry cells in humans. You see individual spikes, and you have to sum up what that activity looks like over time across multiple trials in order to see patterns and change. That is how electrophysiological recordings are analyzed. We also have functional magnetic resonance imaging, or fMRI. There's a couple of other different kinds of imaging which we will see across the semester, but last time we talked about that in humans you can see changes in blood flow in this brain area called MT, which is critical for looking at and detecting movement within the visual field. And MT is particularly sensitive to movement. And so for this, you can put a human inside a machine, show them something. So this human would be seeing something, and it's got like a little readout here, pushing some buttons or something. Some kind of task is going on, but they're in this giant fMRI machine, and it can detect changes in blood flow across the entire brain in real time, and it'll look at changes activity happening on the order of seconds. So it doesn't provide quite the resolution that the electrophysiological recording does of an individual neuron or like a handful of neurons, but it looks at big brain areas across time, but it's a very non-invasive way to do this, and it's oftentimes the only way that we can get any recording of activity in humans, and sometimes this is also done in animals as well. So those are two ways of looking at measuring neural activity in live animals. I just want to emphasize these things because it's really critical that you know what these different kinds of techniques are. So we're going to talk about another way of measuring neural activity in live animals. And this is becoming a more and more common way of doing this kind of work. And it's really kind of exciting and cutting edge, almost science fiction-like. And it uses, it takes advantage of the fact that there's a change in intracellular calcium that occurs when a neuron fires. So a neuron at rest will have a certain relatively low level of intracellular calcium. There's intracellular stores of calcium, like in the endoplasmic reticulum, and then you have calcium channels that are closed and pumps that are continuing to push out calcium out of the cell. So intracellularly, it's relatively low. Calcium concentration is low at rest. As soon as there's an action potential, the calcium channels open, and there can be release of calcium stores happening from the endoplasmic reticulum. There will be an increase of calcium concentrations within the cytoplasm of a cell. So that will go up alongside an action potential. So we're looking at membrane voltage here. Potential goes up, it's depolarized after hyperpolarization. But over this whole time course, we have this sharp increase in calcium, and then it slowly goes down over time. And by slowly, I mean on the order of 100 to 200 milliseconds compared to an action potential. So we have all this calcium within the cell, and over the last 15 years or so, scientists have been using genetically modified green fluorescent protein to detect changes in intracellular calcium. So what the most commonly used genetic tool that's done for calcium, doing calcium imaging is called GCamp, and it can detect an increase in calcium. So if calcium increases, what will happen is that one domain of this will then interact with calmodulin. Calmodulin is a calcium-sensitive protein. It will take on the excess calcium, causes this protein to translocate here, which allows this green fluorescent protein to glow when you shine a light on it. But if there's no calcium, this protein is now dissociated from the calmodulin, and you can't get it to glow. It doesn't glow very well at all. So you can detect calcium. Calcium will be present by looking at just how much the cells are glowing. And the change can happen very, very quickly. So that's shown here, in fact. So calcium imaging can be used to monitor activity in neural populations in whole animals. So you can make a genetic mouse or transfect a mouse using viruses. You can inject it into a particular brain area, causing the neurons in that area then to express this GCamp calcium indicator. So the cells will start expressing this protein, and then you can make a little window on the surface of the mouse's head and lower a microscope onto its skull, and then record using a microscope, looking at the fluorescence of cells within that area. And so at any one time, you might see a little bit of fluorescence, but as a population of cells gets activated, you will see this increase in fluorescence, which will be driven by the influx of calcium happening at that time. So what you're seeing here is just a repeated image over and over again of neurons that have relatively low calcium, and then some changes happen. Say the animal has smelled something, and then a subset of all those neurons are now having an increase of intracellular calcium, and you can see this sharp increase in fluorescence. And so you can see that they fired, and you can see which ones fired. So this has been done over and over again in the olfactory system across a bunch of different labs in mice. And what we've discovered is that if you're looking at a particular part of cortex, piriform cortex has been the most heavily studied, but you can see similar things happening in the entorhinal cortex. What happens is that when you're imaging in that area and you put on, say, one odor, you will see a subset of the neurons fire. So that's green here. So this is looking at alpha pinene, which gives a kind of piney smell as far as we perceive. Those green neurons are firing here. So, and you can see them all firing all at the same time using this calcium indicator technique. So all at once, you can see a big a subset of the population firing. Then in the next 10 seconds later, you can then puff on octanol, which gives a fruity or citrus smell, and then you'll have a different subset of neurons that fire. So then these neurons are firing, and they're showing the red ones fire. You can see some of them are yellow, and that's because that subset of neurons fired for the alpha pinene and the octanol. So it's the again, more population coding that's happening in these areas of the olfactory cortex that's giving you the ability to detect pinene or octanol or both. So let's say then we do the next one. So in the next phase, comparing these two different odors, we have hexanol. So this is the smell that gives you cut green grass. And that's going to give us a green color here. So we're using green for that. So that's a different subset compared to the ones here. But in this same animal, we do octanol again. We're going to see red, and it's going to be the same red ones. If you go from here to here, you can see it's the same red ones here, 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 there. Same red ones because it's the same odor, but the green one is different. And so you see different neurons responding to this odor than this because it's a different set of olfactory receptor neurons that have been activated. But again, there is some overlap. Some of these neurons are receiving octanol information and hexanol information, but they're not receiving necessarily the same neuron information from the alpha pinene. So here we're starting to get some convergence of this information, and it's the combination of this information that allows us to tell whether we're talking about hexanol or octanol, and it's that population coding activity, and it's that constellation of neurons that fire upon odor exposure that gives us the salience of which odor is it being expressed which, and allows us to tell and create memories of the odorant context, which odors were present at a particular time, feeding forward into the hippocampus and the amygdala, and allowing us to connect particular events to odorant experiences. And that's how that's done from a population coding perspective. So in this slide, I'm just going to briefly go over the 4 basic steps that all sensory pathways have with respect to olfaction. We have sensory reception. We have odorants coming into the nose, and they are sensed by the olfactory receptors that are located on the cilia of olfactory receptor neurons in the olfactory epithelium. That induces a change in the membrane potential. So there's transduction of that chemical signal into the kind of information that the nervous system can then read out. So that there'll be changes at the membrane potential, those cells depolarize and then start to fire. And then there's transmission of that signal where the information is gathered into specific glomeruli that will have odorant information, inducing firing in mitral cells that then is transmitted up to the cortex, where there is processing of that information so that it can be perceived. And we talked about what happens on the population level within the cortex with populations of neurons firing with specific odorants, and then a different odorant might have a different population of neurons that will fire, but there may even be some overlap depending upon the odorant. So to wrap up, what are the 5 major tastes? How are taste cells organized anatomically? How are they connected in the papillae? What does the taste bud look like? And then you also should know the key molecular events that underlie taste perception in each kind of taste cell. Which cranial nerves are involved in relaying taste information to the brain? What are the ascending pathways as well? What are the olfactory receptor neurons and how are they organized? What are glomeruli and how are they organized? What is the basic circuit and how are they connected up to the olfactory bulb? And then what brain areas are targets of mitral cells and what particular areas are not targets? And there's one more thing that I would add to that, I suppose, which is have some familiarity with these concepts of population coding and have some understanding of how we record and understand neural activity in live animals, and because we're going to build upon these concepts as well. All right, so that's it for this lecture. We'll discuss this more at the appropriate discussion section. The one last thing that I want to just add to this is this is just a helpful summary of some of these different the different taste receptors. So it's a lot of detail here, but this can be a useful summary for studying. So so I included this as well. It has a little bit more detail than you might need to know. So everything that I discussed before in the lecture is going to be the key parts that you'll need to know for, say, exams, quizzes, that kind of thing. But this covers almost all of it. So I still think it's a useful table. All right. Thank you very much.